Hi, good evening. Of course, no one are there. Good evening, who's there in the YouTube also. Okay, I hope every one of you have written the test yesterday, right? So, though that question paper we are going to discuss today. Okay, just a minute. Before going to that, I just want to check a technical poll whether you all can hear me properly and whether the screen is visible properly or not. Okay, so if any problems are there, I mean, during audibility or visibility of the class, you can just let me know in between. So I hope everything is fine. Are we good to move, good to go to the questions? Yes or no? I'm waiting for the answers. So, yep. So today what we are going to discuss is that the question paper the test paper which you have written the test on yesterday. So that is on the 16th April. So the test you have written on the 16th April and the solutions we will be discussing on the 17th April. That's it today. Okay. So without wasting your time, let me go to the first question. The first question being here is that yeast is used in the production of what? So any guesses? If you can guess, just guess me. Guess the answers. Meanwhile, I'll read the options. Okay. So yeast is used in the production of what? Citric acid and lactic acid, lipase and pectinase, bread and a beer, cheese and a butter. So can anyone answer for this? I know it's not good to launch a poll because very less, or I can say 0 students are present in the class. So I just want to explain the question here. Yeast is used in the production of what? We all know in the bakery, as we all eat so many items like a bread, cakes, puffs and everything, and along with that beverages like a beer, wines, and all these are produced from what? Yes, you're absolutely correct. These are all produced from the yeast. So yeast is used in the production of bread and beer. So this is very easy question, but I just want to tell you here about the yeast. That is why I have kept this question here. Okay, so yeast belongs to which kingdom? Yeast belongs to which kingdom? That's the question. So this belongs to the kingdom fungi. Okay, so yeast belongs to kingdom fungi. And also one more thing here I would like to mention here is that we all know from the kingdom fungi, like a kingdom fungi, kingdom plantae and kingdom animalia, all the organisms are the multicellular organisms. But here one exception is unicellular fungi. Clear? So yeast, which belongs to the kingdom fungi and unicellular. And one more point which I want to mention here is that in the fungi we have a different classes also, different groups of fungi, right? So this belongs to the Ascomycetes. Have you heard about Ascomycetes, Basidiomycetes? If you have a doubt, just put the doubts in the chat box so that I can clear the doubts. Okay. So this is what I want to mention about the yeast here, so that in the further you should not get confused. Yeast belongs to which kingdom? Why the yeast is? Why yeast is a special in a kingdom fungi? Because yeast is the only fungi which is a unicellular, but all other fungi are multicellular. This point you have to remember, very much important. And yeast belongs to which group of fungi? That is the Ascomycetes. Okay, so the continue to the same question, like same kind of question. I just want to ask one more question with the same base. So match the column 1 with the column 2. Just now I told you, we must know the organism belongs to which group or which kingdom. The same question here asking there. Rhizopus, column 1 contains Rhizopus, Penicillium, Eustylago and Alternaria, whereas column 2 contains Ascomycetes, Basidiomycetes, Deuteromycetes and Phycomycetes. We know what are these Ascomycetes, Basidiomycetes, Deuteromycetes and Phycomycetes. Can anyone tell? Come on. It's your turn to reply in the classroom, not to sit simply. These are all the group of fungi. Yes or no? Yes. So for each group of fungi in the column 2 contains all the groups of fungi, and column 1 contains, in the column 1 we have the examples of these groups. So only thing we have to do here is that which fungi belongs to which group. Easy. Okay. Yeah. So Rhizopus. Do you have any idea Rhizopus belongs to which group of Rhizopus belongs to which group of fungi? Yes, Rhizopus belongs to the Phycomycetes, and Penicillium belongs to, and Eustilago is a example of Basidiomycetes, and Alternaria is an example of Deuteromycetes. So can anyone tell me out of these 4 which is called as a fungi imperfect? Just along with the questions, we'll read a 1 or 2 words extra also, so that nothing is going to lose for us, right? This Deuteromycetes is called as fungi imperfect. Okay. So why, ma'am? Why we are calling this as a fungi imperfectly? Why? Because in the Ascomycetes, Basidiomycetes, we'll know what kind of sexual reproduction is going to happen and what are the Phycomycetes, we'll know. But there are few fungi, fungal organisms, those sexual reproduction, type of sexual reproduction is still unknown. So all those are included under Deuteromycetes. Okay, so these Deuteromycetes fungi we do not know, actually we do not know the type of or mode of sexual reproduction. That is why these are called as a fungi imperfect. Okay, let's leave that. Let's come back to our options. So our option must contain A is Phycomycetes. Okay, if there is one advantage of the match the following. You know what? Here we should not know all the 4. Any 2 we know perfectly, then that's fine. Let's say I know about a Penicillium which belongs to Ascomycetes, and I also know the Phycomycetes. Rhizopus which belongs to Phycomycetes. So first let me say for A it must be S. So for A it must be S. Option A is having, option B do not have, option C is having, but option D do not have. So only because of 1 here I can eliminate 2 options, right? Next, I told you I know about a Penicillium also. Penicillium which belongs to Ascomycetes, which means only one thing we have to see in both of these, A and C. Penicillium means for the B it is a P. For the B it is a P. Yes or no? Yes, that is in the option C. So option A is also not the correct one. Here option C is correct. So, mostly what I prefer and I can suggest to you is that whenever we are going for the match the following, instead of choosing, finding the correct option, correct answer, just see in which you are perfect and eliminate the other options. That is a perfect way and easy method to find out the answers in the match the following. Yes, I hope you all can follow me what I'm saying. If you have any doubts, yes, I know you are going to ask in the chat box. Next question. In which plastids are not found? Blue-green algae, bacteria, fungi, and all of the above. Can you give me the answers? Any answers in the chat box? Any answers in the YouTube? If in case, okay, let me know, Manjula. Okay, so in which plastids are not found? First of all, before going to the option, let me tell you what is a plastid. Okay, so the plastid is double membrane bounded structure, which is double membrane bounded structure. Okay, plastid is a double membrane bounded structure, right? So here we have two types of cells. What are those two types of cells? That is prokaryotic cell and eukaryotic cell. Sorry for my handwriting. It's been a long time that I didn't write by using the writing tab, so my handwriting is little not good. Okay. So yes, we have a prokaryotic cell and eukaryotic cell. Why I'm saying this, I'll tell you. So what is the main difference between a prokaryotic and eukaryotic cells is that if we know prokaryotic cells do not have any membrane bounded cell organelles in it. Do not have any membrane bounded cell organelles. But eukaryotes have both membrane bounded cell organelle and non-membrane bounded cell organelle. What are these cell organelles? Both are present in the eukaryotes. So here, let's come back to the plastid. Plastid is a double membrane bounded structure. Yes or no? Yes. So obviously the plastid will not be present in a prokaryotic cells. So it is present only in a eukaryotic cell. This is one way where we can choose our option. Okay. So here bacteria. Bacteria belongs to which kingdom? Bacteria belongs to kingdom Monera. So kingdom Monera, I mean all the organisms which are present in the kingdom Monera are prokaryotes. Right. So which means this bacteria do not have a plastid because that is a membrane bounded cell organelle. Even blue-green algae, also called as a cyanobacteria, they are also belongs to kingdom Monera, which means they are also the prokaryotic organism. So they also do not have the plastids. And one more option we have is the option D. If there is no option D, there is no option A. How can we choose all of the above, right? So now only option which is left with us is a fungi. Even though we do not know what is actual function of a plastid and whether the plastid will be present in a fungi or not, we do not know actually. Okay, if we know, then that is well and good. Directly we can take that a plastid will be present in a fungi. But if we don't know, I'm just saying why I'm elaborating the answers, because if we don't know this, how to approach, that's it. Okay, so now we don't know in which organism we have. I mean, in which organism we have a plastid. So how to approach the question there? How to approach is like the same way. Plastid, okay, which is double membrane bounded structure, we know that, or cell organelle, we know that. So this is present in only eukaryotes. It is not present in the prokaryotes. So now let us eliminate the eukaryotic organisms. If we have a eukaryotic organism, we can eliminate. Or let's say all the prokaryotic organisms are only present. Then we can choose all of the above. Okay, this is a one way, I mean, another way of approaching the answers. I mean, another way of approaching the question. Clear? Any doubts? I guess no doubts. Next question is, which of the following is related to glycosylation of protein? Glycosylation means, just a minute. Okay, so glycosylation means where the ribosomes will be getting converted as a glycoproteins. Glycoproteins. Okay, so which of the following is related to glycosylation of protein or glycoproteins? How do they form? Option A is by endoplasmic reticulum, option B is a peroxisome, and option C is a lysosome, and option D is mitochondria. And we know that mitochondria is not the answer, because mitochondria is mainly for the production of ATP. This is to produce a ATP. Okay. and not the peroxisome also, and not the lysosome also. But lysosome, we know that suicidal bag, right? So which is the answer here is that endoplasmic reticulum. If you know that GERL, do you know this? The girl, right? So which means what? G for Golgi apparatus, ER for the endoplasmic reticulum, and L for lysosome. First, the proteins or the lipids, whatever it is, it starts to form in the Golgi complex, and then they move to the endoplasmic reticulum. Even the protein also, which starts producing in the Golgi complex, and then it packed in the endoplasmic reticulum as a glycoprotein. Okay, this is called a glycosylation. Next question here. What is the next question? We have is, starch and cellulose are the compounds made up of many units of what? Starch and cellulose are the compounds made up of many units of simple sugar, fatty acid, glycerol, and amino acid. Here we'll be getting 1 minute to think and say the answer. Yes, done. I hope you got the answer. Starch and cellulose. Okay, so let me give the answer to this. What do you mean by starch and cellulose? Starch and cellulose. Starch and cellulose both are the polysaccharides. Polysaccharides. What do you mean by polysaccharides, ma'am? Okay, I'll tell you. First, let me tell you about the monosaccharides. Only one unit will be there, like a sugar. So if 1 unit of sugar, normally it will be having a CN, H2N, ON. Okay, CN, H2N, and ON. This is a 1 unit of a sugar. This is called as a monosaccharide. Because only 1 unit we have, whatever the number of N, we are not, we are not bothering about it. Okay. Monosacharide. And next, there are other sugars which are having a CN, H2N, ON. And to this, the bond is going to form. And again, CN, H2N, ON. So now how many units are there here? 2. So this is called a disaccharide. Now, so you can guess the answer. What is a polysaccharide, right? Polysaccharide which is having a more than 10 units. More than 10 units. Okay, this is a polysaccharide. Like starch and cellulose both are having a more than 10 units of a glucose. Whatever it may be, a more than 1 unit of monosaccharides are there. So that is why the starch and cellulose are called as a polysaccharide. Of course, we have a structural one more point I want to add here is that the starch is called as storage polysaccharide. Storage polysaccharide. So why we are calling, polysaccharide is okay, fine, you understood, right? So why, what is the, speciality of calling again as a storage polysaccharide? Why? Because in most of the plants, in all the plants, I can't say most, but I can say all the plants, the food is going to be stored, synthesized in the form of a glucose and transported in the form of a sucrose, and it is stored in the form of a starch. That is why this is called as storage polysaccharide. Okay, next, what about cellulose? Cellulose is called as structural polysaccharide. Structural polysaccharide. Why the cellulose is called as a structural polysaccharide is because the cell wall of the plant cell, the cell wall of a plant cell is made up of a cellulose where the structure is forming there. So that is why the cellulose is called as structural polysaccharide. So this is about a starch and cellulose. Let's come back to our question. What is our question here is that starch and cellulose are the compounds made up of many units of monosaccharides, as I told you already. But these monosaccharides are also called as a simple sugars. Simple sugars. Okay, so now you can easily guess the answer. Simple sugar. And fatty acid, like let me write the other options here. That is, let's make the entire screen colorful. Option B. Let me erase this. Option B, that is a fatty acid, plus a option C, that is a glycerol. Both together forms a lipids. So here there is no question of polysaccharide. And amino acids together later forms a proteins. Okay. So this is a simple basic question. But I just want to give you a brief of starch and cellulose, like saccharides, lipids, carbohydrates, lipids, and proteins. That's why I just choose this question. Okay, next question we have here in the same way, that is which of the following enzyme was first isolated and purified in the form of a crystal? We know following all the four options are the enzymes. But out of these four enzymes, which is isolated and crystallized, purified in the form of crystals first? Amylase, urease, ribonuclease, and a peptide. You just think, meanwhile, I'll have the water. Where did I get? Yes, please do think for this, and I'll come back to you. Okay. Yes. Did you got the answer? So that is, let me tell you the answer. The answer is urease. Urease was the first enzyme which was isolated and purified in the form of crystal. But first means when? In the year of 1926. In the year of 1926, this urease was isolated and purified in the form of a crystal by a scientist called J.B. Sumner, okay, whatever. J.B. What do you pronounce it? Sumner or Sumner? Okay. So he was the one who first isolated in the year of 1926, isolated and crystallized the urease enzyme. And after that, all the enzymes we are followed the same way. Okay. Next question here is the most important property of an enzyme it is. What is the most important property? Composition, thermal denaturation, specificity, solubility, which is the one? Here looks like all the four options are the properties of enzymes, right? But what we have to do is that we have to identify the most appropriate one. Always you might have seen some of the memes like the same picture on 4 images. I mean, same image in the 4 options, and they'll ask you find the odd one. Sometimes our questions will be also like the same, because all the 4 options looks the same. All the 4 options are feels like, okay, this is the answer, this is the answer, this is the answer. But when we go in depth, then we'll understand, I mean, we'll get to know this is what the actual answer. So here also composition is also, I mean, the property, I mean, one of the important character of the enzyme, and thermal denaturation. If the temperature is increasing more and more, the enzyme, which is a protein, will get denatured, right? And even the solubility, everything, specificity, everything are the properties. But which we have to consider as the most important one here is the sorry, that is as a specificity. Why we are considering this as a specificity? Because a particular enzyme will be acting on the particular substrate. You can remember the lock and key theory of enzymes, right? Substrate. So this is why this is, I mean, specificity of the enzyme is the most important property. And let's say thermal denaturation, it's not, yes, it is important, but not the most important again, because it's not the most, because all the enzymes, if the temperature has been increased, all the enzymes will get denatured. I mean, there is a particular proper, I mean, temperature, optimum temperature for all the enzymes, but the specificity is not same, because if we have an enzyme A, it is going to work on the substrate A only. If we have an enzyme B, that is not going to work on a substrate A. It will be working on the substrate B. So specificity of the enzyme will be considering as the most important property. Clear? I have got a so good students that you have no doubts at all. Okay, so next question. So fastly I'm moving to the next question. I hope you all will be asking the question so that the session may not be completed in 1 hour, but maybe time may get remain. I don't know. Let's see. Mark the correct sequence of a steps for the development of a root nodules in a legume. So here we are studying about a symbiotic association. Symbiotic association. Okay. So in this symbiotic association of rhizobium and the leguminous plants, that they're asking how the root nodule will be formed in those leguminous plants. So we know, I mean, let's read the statements and then we'll make them proper arrangement. Okay. So first one is formation of infection thread, division of cortical and pericycle cells, and that is and pericyclic cells, curling of root hair, leg hemoglobin synthesis. So which is the first step here? Can you tell me? So as soon as the bacteria enters, the first step is that the root hair will get curled. Okay, so if this is the root hair, this will get curled first. Okay, so first root hair curling, and now I mean that all the bacteria obviously first will come and then root hair will get curled, and after that what is going to happen here is that just bear with me, I'm very bad at drawing. Okay, so let me draw the bacteria with the red color. So now what happens, this bacteria starts to form an infection thread. Yes or no? So now formation of infection thread is a second point. What is the next? What is the next one here is that obviously once it crosses the epidermal cell, then we have here are just a minute. Yes, here we have all the cortical cells and the pericycle cells. Cortical cells and pericycle cells. So now what is going to happen? The third one is that division of this cortical and pericycle. So this infectious thread will reach the cortical cells and the pericycle cells, so that division will take place. And ultimately leg hemoglobin, which is a pink color pigment. What is the importance? Yeah, leg hemoglobin will get synthesized. Why this leg hemoglobin will get, should synthesize? Because this leg hemoglobin, which is acting as an oxygen scavenger. You might be knowing this, right? So oxygen scavenger, why? Because if the enzymes which are present in the bacteria should work, they will be working in the absence of oxygen. They are anaerobic. So to provide the anaerobic condition, the leguminous plants will be having a leg hemoglobin pigment, okay, which is acting as an oxygen scavenger. So now we have got, first is a third one. So let's see, first is a third one. How many are have? Only C and D. So I'm eliminating A and B. Next, second one is a 1. Obviously, we have option D, not the option C. And then 2, let's cross check. Then 2 and then 4. Correct? Are we correct? Yes, we are correct. In this question, I just want to tell you here we do not have X, Y, Z. So this is what the order X, Y, Z. The diagram below shows the plant with a identical leaf surface area. So they have, all the plants have the same leaf surface area, but the conditions provided here are the change. Like in the X, we have a 10 degrees Celsius of temperature and daylight, normal light with completely, and 20 degree Celsius we are providing for the Y plant with the red light, and 30 degrees Celsius for the Z plant with the green light. So assuming all other conditions were identical for all 3 plants, which of the plant would likely to photosynthesis slowest and which is the fastest? So for this, we must know the factors which are affecting the photosynthesis. So the one factor is that the light, which light will be affecting here. Okay, so we all know red light, what is that? Immersion effect. Do you all remember the immersion effect? If you can remember, just recall the immersion effect. This is what a homework for today, okay? Recall the immersion effect. All the historical background of photosynthesis, that is important. So there will be coming to one conclusion that red light is the one which is going to be stimulates a more photosynthesis. 1. And temperature must be optimum. Always the temperature must be optimum. It should not be low. It should not be high. So here we can consider 20 degrees Celsius is a optimum temperature. So the Y is the fastest here, right? And one more, in the green light, no photosynthesis is going to happen. Daylight, which is a mixture of all the lights, all the colors of light. So the average temperature is also there. So average photosynthesis is going to happen in the X plant. In the Y plant, the maximum photosynthesis, the fastest photosynthesis is going to happen. Whereas in the Z plant, as the plant is having the green light, the photosynthesis, I can't say the photosynthesis is 0, but the photosynthesis is very slow. So that is why in the Z, the photosynthesis is slowest, and in the Y, the photosynthesis is the fastest. Is it clear? And X is the intermediate one. Clear? You will not respond. Okay. So match the following. 116th question. Directly, I'll give you the answers here. For the auxin, it is a xylem differentiation. That's a cell differentiation for the auxin. And for a cytokinin, it is a nutrient metabolism will be depend on the cytokinin. These are all the hormones, and column 1 contains the hormones and column 2 contains some functions. Okay, and ethylene is root hair formation, and abscisic acid will be helped in the seed development. So here the mostly I know is a abscisic acid, ABA, which is for the Q. But D, Q, how many are there? Only one is there. Done. So only with the one option I can choose my answer now. Right? So next 126, the question, the same we have the match the following. Column 1 contains all the nerves names and column 2 contains how many pairs? Do you all remember the spinal cord? Spinal cord, how many bones we have here, and lumbar bone, cervical bone, thoracic bone. So one good thing we know here is that, listen. One good thing we all know here is thoracic bones. Okay, thoracic bones will know. Thoracic bones means the thoracic area, upper part of our body, where the lungs are present. So thoracic area, in the front side, we have, what do you call, ribs. Sorry, we have a ribs, right? How many pairs of ribs we have? 12 pairs of ribs. The same way, if we go back side, we have a thoracic bones in the spinal cord. Same, 12 pairs. How we have in the front, we have the same way in the back side. So now you can easily guess the thoracic nerves. How many thoracic nerves will be present? Obviously 12. In the thoracic area, everything is a 12. Ribs are 12, thoracic bones are 12, and thoracic nerves are also 12. Okay. So thoracic nerve is a 12 pairs. So now for option B, it is a R. How many we have? Only one we have. Yes or no? So easy. But let's cross check the other also. Cervical nerves. Cervical nerves are, actually we have a 7 cervical bones, but these 7 cervical bones are connected with the 8 pairs of a cervical nerves. Okay, so for A it is S, correct. And lumbar region, in the lumbar region, that is just below the thoracic bones, we have a lumbar whatever, lumbar bones, which are 5 pairs of bones, right? So these obviously have the 5 pairs of nerves. So lumbar nerves are 5 in number. And coccyx bone, tailbone, right? So this also is having only 1 tailbone and 1 pair of nerve. That's it. So for the D, it is Q, and for the C, it is a P. Is it clear? Some of the statements regarding the sexual reproduction are given below. Did I take the answer? Yes. Some of the statements regarding a sexual reproduction are given below. So we have to read the sentence and choose the correct statement from the given alternatives. First, let us read the sentence. The same way we'll eliminate the sentence which is wrong. Okay. Oh, yes, we have to choose the correct one, right? Let's choose the correct one. First one, sexual reproduction does not always require 2 organisms. Choose the correct one. Sexual reproduction does not always require 2 organisms. This is one first point. And reproduction usually involves a gamete fusion. There is never meiosis during a sexual reproduction. External fertilization during a sexual reproduction is a rule. Okay, so In human, it is internal fertilization. Then how can the sexual reproduction, I mean, external fertilization is a rule in sexual reproduction, right? And to form the gametes, meiosis must be there. So meiosis during sexual reproduction is required. And reproduction usually involves in a gametic fusion, yes. And sexual reproduction, it does, I mean, it's not, let me think of one example. Yes, I'm thinking of one example where sexual reproduction does not always require a two organism, not always, but let me give you the example for this later. But for now, you take this as option 1 and 2. This I'll note down. Okay, I'll just give you the examples for this. Next question, 133. Most of the sexually transmitted diseases are completely curable if detected early and treated properly, except except this, the remaining we can treat. Except this, except hepatitis, except genital herpes, except HIV infection, or except all of this. Which is our answer? Sexually transmitted diseases are so dangerous that where we cannot even detect during the early stage. Okay. So all of these is the answer for this.